Now here comes the role of a pharma drug. When we talk of pharma drug, a molecule, chemical, whatever molecule you are going to use, we cannot change the homeostasis of the body. That drug molecule will never change the functional status of the cell. In whatever way it may be, it will neither help to regenerate the cell nor to improve function of a cell. This is very important. Why I'm insisting this hypothesis is that, and new drug molecule is that, when we talk of a new drug molecule, we need a molecule which can change the deregulation into regulation. We need a molecule which can bring the ECM and cell dysfunctions into normal functions. And none of the drug molecule can bring this. Whatever we do our research of whatever volume, none of our pharma molecule or chemical can do this change. So we need a molecule which can integral part of this system, which will become a part of the system and change the milieu or environment where the cells become functionally regulated. So that is a new horizon of drug. What I'm talking is that's a new horizon of drug. This new horizon of drug is a living drug, an entity which you can say universal molecule which can treat multiple diseases and one cell. Whereas a pharma drug, one cell, one disease, and that to one symptom, not multiple symptoms, and that too with related toxicity, and that too related with the complications of the drug, and ultimately the patient succumbs of the complications. This is what I was talking about non-living conventional drug and its limitations. Only alleviate the signs and symptoms. Side effects becomes a major issue there, and actions are very transient. They don't change the disease profile. Ultimately, a disease goes progressive in most of the cases. I have two examples for you which traditionally we have been following. One is insulin, the other is nitroglycerin. Insulin we have been using for hundreds of years, nitroglycerin for 150 years we have been using. None of them can change. Neither the beta cell functionally changes by insulin, nor myocardial cell by nitroglycerin. Years have been practicing. Ultimately, patient doesn't respond to this drug and gets into a complication. So we have to think what is that we can change the system to regulate it back from deregulation. So ultimately, it comes a model of regeneration, that is stem cell. That's what I call a living drug, a molecule which is breathing entirety, which is communicating with the environment around, which is understanding the changes taking place around, and sets the system normal. So we need a need of our change in healthcare system. As I said, normally this is where we fail in the health system, I call as a health crisis. This health crisis is a very specific sentence I use. Its reason being that ultimately, with the most advanced gadgets and investigations and research, we have given up the treatment for most of the diseases which are enlisted in the form of chronic debilitating disease, aging, cancer, even diabetes. We are unable to do anything. So I call them as a health crisis. We are at the end where we cannot do anything. So we need to think parallel. What is that is very important. Innovations are required, new treatment therapies are required. We can't solve the problem by using the same kind of thinking we used when we created them. This indicates that whatever efforts we do, whatever amount of energies and finances we put in the research in a conventional method, we are not going to get solutions to the problem. So we need to change our parallel thinking, and this comes from a disruptive innovation, and innovation above a proven one. When we talk about proven therapies, means proven therapies are which we are practicing as a physician. I'm a clinician, I'm basically by training a neurosurgeon. Means as a neurosurgeon, I have noted my own limitations where I have to stop my practice. And where I say patient, yes, I cannot do, you can. So when you want to go above this, we have to prove the things above proven things, and then we come to closer to the nature, which is the evolving regenerative medicine science. So as I was talking to you, it's a broad spectrum umbrella covering a lot of molecules, cells, helping in rejuvenation, restoration, and regeneration. Whenever new science comes in, the three requirements come as a basic requirement. A research with the researchers, wealth and finances and industries required in it, which has to take a motivation to participate in this research, and most important, politics means regulatory authorities to regulate the new science. If these three sciences put together, I think MSCs can come as a universal molecule for many of the conditions where we think where the conventional methods have failed. Their unmatched qualities as a stem cell in situ, differential potential, paracrine cells, paracrine effect. Paracrine effect is a very big one. Hundreds of paracrine effects are there. As an experimental model, as a raw material for particularly tissue engineering. I call them, subsequently in one of the slides I'll show you what I call them. I call biological plastics. If you remember 1960s era, where the revolution of plastic revolution came in. Dustin Hoffman movie was there, and everything was about plastic. Fountain pen to radios to purses to everything was plastic. And now, after 50 years, I look, this is a biological plastic solution, exact 50 years to 200 time, and we are looking at a molecule which is a biological plastic, which we can mold to any type of cell, any type of organ, one single cell, and a scaffold, a single cell, and different medias, and you find different cells to multiply. The plasticity is very high. The only what challenges in this are going to be there, how do we scale of this manufacturing when we talk of as a drug, as a universal drug. This is what the plasticity of mesenchymal stem cell. If you see, it has a potential to differentiate whatever you talk like dedifferentiation, transdifferentiation, and differentiation into also multiplication. You can multiply, you can differentiate any other tissues. Basic MSC characteristic, if you see, chondrogenic, osteogenic, adipogenic, as well neurogenic. Then you can, with manipulation, you can differentiate into any type of cell like. The chance hypothesis for parasite, whenever there is an injury or whatever kind of injury, MSCs respond in a very different way. As I said, it is a living, breathing entity communicating with the environment around. What is it speaks? When it communicates with the environment around, it senses what is required there. Accordingly, trophic factors are released, unconditional. That's a 24-hour pharmacy of your body. Whether you know or don't know, it's a 24 hours functional pharmacy in your body. It is releasing all the trophic factors. You can say anti-scarring, anti-apoptic, angiogenic, mitogenic, immunomodulatory, antimicrobial is very important. LL37 IDO is a very important molecule which is also antimicrobial. If you see overall, the MSC having a differentiation potential, one part of it helps into regeneration in the in vivo as well as in vitro using biological scaffold. Whereas within the body, if you see, depending on the condition, autoimmune condition, immunomodulatory plays a major role. The same cell, but the molecule is different. Same cell, pro-inflammatory, molecule is different. Same cell, but the molecule is different, anti-inflammatory. Same cell, antimicrobial. You can imagine the plasticity of the cell. That's why I call it biological plastics. It speaks the plasticity of the cell to use it in the therapeutic application, where we even cannot imagine how wonderful this cell could be there as a universal drug for all of us.